So today we'll present this much smaller research group. You get to hear from all of us. So not me or just Marie if you don't speak French. And there's Sarah Dyer-McComb, a master's student in zoology, and Ron Tuganov, a PhD student also in zoology. And I'm cross-pointed in statistics, and most of my work involved developing models to understand the ecology of aquatic species. And in particular, what we'll be doing here at UBC is trying to understand the effect of anthropogenic changes on the population size, the survival, the health, and the reproduction of aquatic species. We have pretty broad interests, so we're interested in marine and freshwater fish, and seabirds and marine mammals, and similarly we're interested in a range of human pressures going from climate change to fishing, but things like also the effect of dams. And in general, we use two types of data. So we use, and my specialty is movement and space use data, and the idea is that you can understand how anthropogenic change affects species through movement, because a lot of animals change their distribution when there's increased human activities or change their behavior, and you can detect that change in behavior through changes in their movement patterns. And then the effect of anthropogenic change on population is more classically studied through changes in population size, and a lot of people do this here, and that's an approach that we're also going to take. And using these two types of data is important for conservation. So first, it's important to look at the space use and movement of animals, because there's a lot of legislation in Canada and abroad, such as the Species at Risk Act, that says that a key aspect of an animal protection is the protection of its critical habitat. So understanding how anthropogenic change affects the space use and habitat use of animal is kind of the first step towards identifying critical habitat to protect. And then of course, if you're managing a population that's hunted or fished, you want to understand how fishing, for example, affect the trends in population size to better manage. So, as a lot of you know, it's difficult to understand the effect of human pressures on aquatic species, because the data for aquatic species is pretty messy and challenging. So the animals live underwater, or they live in the high seas, so it's very hard to observe. So we get data that's difficult to tackle, and that's where the statistics comes in. And as a research group, we're focused on a general statistical framework, which is the use and the development of spatial and or temporal hierarchical models. And one of the key aspects of hierarchical model, which I'm sure a lot of you actually use in your own research, is that you have a hierarchy of equation. So you have, at the top level, an equation that links observation to the underlying mechanism. And the idea here is it takes into account the fact that the data that you're gathering is not perfect. So whether it's messy movement data or very messy population data. And then the second level explains the mechanism that we're interested in studying. So for example, here if we're looking at fisheries data, the measurement equation would model how fishery landings are gathered, for example, and then the process equation would model how the population changed through time. And a big focus of our group is to develop more mechanistic model that explains more the details of these processes, and in particular by including things like human activities or their effect directly into the models. And then when we're Bayesian, which is not all the time, but sometimes, you have another level, which I'm not putting here, but that explains the uncertainty in the parameters of the model. So there's three teams that we'll focus on in the next few years and that we'll talk about today. The first one is to link movement to its population and fitness consequences. And the idea is to develop movement models that explains the movement of the animal based on environmental covariates, including human activities. But the key aspect that makes this work novel is that we will also link the movement to fitness proxy and population level data. And a lot of the work that's done in movement ecology now is really just focused on describing the pattern of space use. And by linking that movement to the effect that it has on the animal, the population size or individual size, we can really understand the consequences of movement on the animal's survival and reproduction. The second team is to develop methods for novel aquatic data. So there's been a lot of work on GPS tracking data, which is kind of the focus of the first team, but there's not a lot of sophisticated models for data that's gathered with things like acoustic receivers. And developing models for this type of data is really important because a lot of species that live underwater can only be tracked with these kind of receivers. So for example, all the fish that don't surface, so most of them, cannot be tracked by GPS. It needs to be tracked with things like acoustic receivers. So by developing methods for this novel type of data, we're kind of developing one new way to understand how anthropogenic exchange affect their space use in finding ways to identify critical habitat. And then the last theme that I'm going to work on is improving stock assessment model, and that's definitely a field that there's been a lot of very sophisticated methods that have been developed, including by people here in the IOF. But there's still, I think, place for improvement, and in particular, there's models that are used in management currently that I think we can improve. So I'll be working with the eco-scientists to develop a set of models to really take advantage to the data that's available to its full extent. And with this, I'll pass the torch.
So I'll talk a bit about the first thing, which is to link movement data with the fitness and population consequences. And underlying the link between these is the fact that movement data that we observe reflects underlying behaviors that have certain energetic consequences because they're associated with energy intake and energy output. And to link the two of these things, it's a multi-step process. The first thing you have to do is, based on the movement data, is identify a behavior of interest. For example, foraging. Then you could either directly model the effects on the population, or on survival, health and reproduction, which Sarah will talk about for her project, or you can do it indirectly. So if you have a behavior that you know has fitness consequences, like foraging, you can identify the environmental conditions that are necessary for that behavior. And then if these conditions change, you can infer how that might impact this behavior that has those fitness consequences. So if the conditions required for foraging become less frequent, you can infer that either foraging will also become less frequent or the behavior will have to change. So between Sarah and I, we're working on 3 species, polar bears, narwhals, and gentoo penguins. And I'll talk about the first 2. And we're both focused on foraging. So polar bears' primary habitat is the sea ice. On the sea ice, they do the majority of their foraging. And because of the huge scale of this habitat and the complexity for them, they rely heavily on olfaction to locate their prey. And one of the main things that affects an olfactory predator is wind. And an animal has 3 ways it can move relative to wind: upwind, downwind, and crosswind. The cone represents the area they perceive by olfaction. Now, moving upwind or downwind only adds the traverse distance to what they perceive. However, moving crosswind adds the much larger area upwind of its whole path. So this is, crosswind movement is what's predicted as the optimal search strategy for an olfactory predator. And it's this rapid crosswind movement that I'll be using as a sign of foraging polar bears. And to identify this, we have GPS tracks of polar bears, and we'll compare these movements to wind data that are used for weather forecast systems. And once movements are identified, they can be modeled based on environmental factors that might affect them. So some of them might include wind speed, which affects the efficiency of prey localization. So once you smell something, how easy it is to locate that prey. Ice concentration and carbonation, which affects prey distribution and the energetics of moving through that habitat. And ice drift, for which polar bears have to compensate to maintain their geographic location. Now, In contrast to polar bears, narwhal foraging has different underlying characteristics. Firstly, narwhals are group animals. They're not solitary like polar bears. The second is that they rely on echolocation to locate their prey at a fine scale. So their ecology, their foraging ecology, would be different. So to identify in narwhals, we have narwhals tagged with a huge suite of biotags, including horizontal fast-lock GPS, vertical dive sensors, high-resolution triaxial accelerometers, AccuSound, and AccuSound hydrophones. And I'll be using a two-step process to study narwhal foraging. The first is identify what foraging looks like using high-resolution data. So using the dive profiles, the triaxial accelerometers, and buzz and click detection from the AccuSound, we can fairly accurately identify foraging attempts. The issue with the high-resolution data is it's very energetically demanding on the biotags, so they have a very short temporal range. So the second step is to link these high-resolution, the foraging identified by the high-resolution to low-resolution data like the GPS and low-resolution dive data. So this would drastically increase the temporal range that we could study foraging in. And similar to polar bears, once we identify foraging, this can be modeled based on environmental factors. So for narwhals, again, we can use ice concentration, and this has important biological relevance to narwhals since they use ice on one hand as a refuge from predators like killer whales. On the other hand, if ice concentration is too high, that could impede access to surface to breathe. I'll also use temperature and salinity, and this has effects on the efficiency of echolocation by affecting sound attenuation. And lastly, industrial activity, which can also interfere with echolocation and other behaviors. Yep. So.
So to continue on with theme one of linking movements to fitness consequences, my project more directly relates them to the animal's fitness. So for this, we're going to talk about the gentoo penguins. And gentoo penguins are interesting because they are facing a lot of anthropogenic threats. There's fishing, they can be caught as bycatch, and there's also a lot of oil exploration in their interest in their habitats. So they do have a lot of human activities in their areas. But aside from these human activities, they also experience severe energy depletions on their reserves during the breeding season and the pre-molt and when they're molting interval period. So because they go through this time where they really use up all their energy, it's crucial for winter foraging that they are able to increase and replenish their energy reserves. So this can be done with varying degree of efficiencies. It can be kind of hard to find prey in their environments. And from this, their movement decisions are really critical in depicting on how successful they are in increasing their energy reserves. So if they're not able to really recover their energy during the winter, which is likely to be the case in some years, as especially in the Falkland Islands, we do see the colony populations fluctuate quite drastically, then you get carryover effects into the next season. So if they're not able to recover their condition enough, then you have impacts on their reproductive readiness into the next season, on how well they're able to invest in their eggs and raise their next batch of young. So you can get a lot of valuable information when you combine movement data with diving profiles. So for gentoo penguins, we're going to attach paired satellite transmitters and time depth recorders, which just came in the mail. We're very excited. And from this, we can kind of use, we can model using their turning frequency rate, their dive depth, and also the wiggles in their dive profiles to identify foraging locations. So this is an example of a dive that there's no wiggles, so we wouldn't assume that that animal's foraging. But in this one, because of the pattern, we can assume that there is. And there's been a lot of work done on a variety of penguin species with video recording and other methods to kind of verify this. So from this, we're going to use state space models to account for different errors in the Argos data, which is the movement data that we're going to be collecting. And then we can also maybe go into switching state space models to really try and tease out the foraging behavior among their just traveling behaviors. So from this, we're also going to be collecting blood samples and feather clippings and some other measures of the animal's reproductive readiness and their fitness. So we can look at body mass, their reproductive physiology, things like egg mass are a great indication of their reproductive investment into the next season. So we're hoping to collect a variety of data. And then we're going to be able to directly relate it to their body condition, relate body condition to things like their foraging efficiency. So this would be the case of how far do they have to travel to get the same amount of foraging events occurring. And we can also look at their area and habitat use. So if individuals have different areas of habitat that they're focusing on, we can kind of look at the different environmental factors taking place, such as sea surface temperature, and also be able to directly link their fitness to anthropogenic effects, such as heavy fishing in one area, where another area doesn't really have it. So on to theme number 2, which is developing methods for novel aquatic spatial data. By this, we're kind of referring to 2 kind of sub-branches. We're going to look at underwater detection data, such as from acoustic tags and receivers, but also opportunistic data. So this could be from old field programs, citizen data, non-systematic boat surveys. So for underwater detection data, you have an acoustic tag that is transmitting an acoustic signal, and it's picked up by receiver hydrophones under the water. And as Marie mentioned earlier, for fish, when you have an animal that lives mostly underwater, you kind of have to get creative in how you're able to detect where they're going and where they're moving in space, because they don't come to the surface. So from this, you can kind of create different arrays of receivers and get a lot of information about where the animal's traveling in their habitats. So this could be set up along riverbeds, rivers, and lakes to try and figure out where animals are in space. So while this method is becoming really common to study fish, but most analysis are just primarily descriptive. So you have identifying species, timing of migrations, and animal counts. But most of these methods are largely ignoring the spatial aspect of their data, which is actually quite a rich resource of information, although it's kind of complex to handle. For this, we're going to adapt spatial capture-recapture models, which have been used in the terrestrial world a lot for those motion-detected cameras that kind of activate and then they get an image of the animal. And then also Gaussian random fields, which is used in fisheries a lot. So we're going to use a case study species with the Arctic grayling, and this is in collaboration with Eduardo Martins. So the Arctic grayling has been negatively affected by dams, and there's a need for conservation, but we don't really have a lot of information on their habitat and where they're abundant. So the goal here is to identify a relationship between density and environmental covariates, and it's unique because we get to kind of plan where we're putting out the arrays and receivers the entire way up. So you can kind of work from the ground up to work out the spatial methods to now analyze it. The opportunistic spatial data, which, as we mentioned, boat surveys, old field programs, and citizen data. So GPS units are increasingly common. Go and most ferry operators will log encounters with whales as they're traveling across passages and stuff. And also you have a lot of data from whale watching. So because all this data is not systematic in their design, it's collected opportunistically, there's certain biases involved with their collection that make it really difficult to associate to different patterns. So for an example, we're going to take data from whale watching. So this is what Marie and I worked on out in Halifax. But you can see from the GPS reference encounters with longfin pilot whales that there's a search effort bias. So in later years, the boats were traveling and searching a lot further offshore than in earlier years. So this actually has doubled with dividing the years this way. And it kind of makes it difficult to model relationships that involve distribution and how they've changed through time. So we see that we have a hotspot detected for resting activity in the chunk of later years, but we don't have any information that far offshore in the earlier years. And additionally, the effort that we're getting is a lot more sparse further away from harbor. So there's certain biases in the data that make it really challenging to adequately describe patterns that you're seeing because of the effect of where the data is distributed. So we're going to develop models to account for the sampling bias. So in summary, for developing methods for novel aquatic spatial data, in underwater detection, we're going to use hierarchical models to deal with its complexity. And for opportunistic data, we're going to make methods to handle the sampling bias. And the goal is to relate, population distribution data to environmental covariates and other anthropogenic threats in the area.
And then briefly to touch on the third theme, which is to improve stock assessments. One of the primary goals of stock assessments is determining maximum sustainable yield, which is the maximum number of fish you can harvest without depleting the population. Now, maximum sustainable yield is largely affected by spawn biomass, which is the number of adult females that are old enough to spawn. Typically, stock assessments use a two-step process to determine spawn biomass, where first they amalgamate many different sources of data into standardized population indices, and then use these to determine spawn biomass. Unfortunately, this two-step process loses sex and age data, and also loses information on this sampling error. And the ultimate effect of this is the predictions of the effects of varying harvest regimes are more volatile and less accurate. So instead, what Marie's lab is going to attempt to do is model spawn biomass directly using the raw data. Now this would retain that sex and age data and hopefully improve the error estimates. And in effect, what this direct mode of modeling would do is see the effects of varying harvest regimes directly based on the data itself, rather than based on the larger number of assumptions that are required for the two-step process.
And just as a note for the stock assessment process, this is specific to a few set of species that we're targeting. There's different methodology in ways to handle stock assessment. But okay, so I hope you got a better feel for what we do in our lab, and please do not hesitate to ask us questions. Lily is particularly insightful, so go ask her. And also, I just wanted to point out that we're growing. So I've been chatting with Marina, who's a new postdoc that's partly working with Evgeny right now, and I've been talking about developing simulation models to test some spatial models. And then I also have a few students that are interested in starting in September. So for example, there's Rowena Griba, and I'm really hoping she's going to join us to do her PhD here. She wants to develop models that look at the movement of Arctic species that incorporate both scientific information with traditional knowledge, which I think is really exciting. Then I have a few stat students that I'm recruiting that will work on the fishery stock assessment and the opportunistic model data that will kind of dig into the details of the statistics and do a bit of statistical theory, which I will spare you today. And then I have a few students that I'm hoping will join IRES program to do more movement-based models similar to what Ron and Sarah are doing. And then that's it.